Good morning, everyone, and thank you for being here today. I'm excited to kick off our presentation on GMP for active pharmaceutical ingredients, or APIs. This is part 4 of our series, and I'll be guiding you through some essential aspects of good manufacturing practices related to APIs. Over the course of this session, we'll explore key guidelines, best practices, and important considerations to ensure quality and compliance in API production. Let's get started and make this a productive and informative session for all of us. Let's now turn our attention to validation, which is a critical component of good manufacturing practice, or GMP, for active pharmaceutical ingredients, or APIs. In this part of the presentation, we'll focus on part 4 of the GMP guidelines related to API validation. Validation essentially ensures that our processes consistently produce quality products that meet predefined standards. It involves a thorough and documented process to confirm that manufacturing procedures, equipment, and systems perform reliably and effectively. This is especially important for APIs, as they are the active components in medications and directly impact patient safety and treatment efficacy. By adhering to validation principles, we minimize risks, ensure compliance with regulatory requirements, and ultimately deliver safe, high-quality pharmaceuticals to the market. So, throughout this section, we'll explore the key validation steps, from process validation to cleaning validation, and discuss how they fit into the broader GMP framework to support product quality and regulatory compliance. Let's talk about validation. First, it's crucial that the policy, intentions, and approach to validation are well documented. This documentation should cover key areas such as production processes, cleaning procedures, analytical methods, in-process control test procedures, and computerized systems. By doing this, we ensure everything is transparent and standardized. Next, we need to identify critical parameters and attributes. These can be determined either during the development stage or based on historical data. Once identified, we define the acceptable ranges for each of these parameters. For example, with active pharmaceutical ingredients, or APIs, we pay special attention to critical product attributes. These are the qualities that directly impact the safety, efficacy, and quality of the API. We also need to consider process parameters that could influence the critical quality attributes, or CQAs. For each of these critical process parameters, or CPPs, we define a specific range. This helps us control the process tightly and ensure consistent quality. Finally, we focus on validation of operations that are critical to the API's quality and purity. This validation ensures that every step in the process maintains the integrity of the API, aligning with GMP standards for active pharmaceutical ingredients. In summary, thorough validation is about documenting our approach, identifying and controlling critical parameters, and validating operations that directly impact quality and purity. This is fundamental to maintaining high standards in pharmaceutical manufacturing. Let's dive into the key aspects of the validation protocol and report. First, the protocol itself must be written, thoroughly reviewed, and officially approved before any validation activities begin. This ensures that everyone is on the same page and that the process is clearly defined. Next, the protocol should specify critical process steps and establish clear acceptance criteria. This helps us determine what constitutes success at each stage. It's also important to specify the type of validation being performed and how many process runs are needed to adequately demonstrate consistency and reliability. When it comes to the report, it should cross-reference the protocol to show what was planned versus what was done. It should also summarize the results obtained, noting any deviations from the protocol, along with appropriate justifications for those deviations. The report concludes with clear conclusions and recommendations based on the validation outcomes. Remember, if there are any variations from the original protocol, they must be documented thoroughly with justified reasons. All of this is crucial to ensure compliance with GMP standards for active pharmaceutical ingredients, or APIs. It's about maintaining quality, consistency, and transparency throughout the validation process. Let's start by talking about qualification, which is a crucial step before we even begin process validation. It's essential to ensure that all critical equipment and ancillary systems, including utilities and facilities, are properly qualified. This helps us confirm that everything is suitable and reliable for manufacturing. Now, moving on to the different stages of qualification. First, we have design qualification, or DQ. This step involves verifying that the design of the equipment and systems meets our process requirements and regulatory standards. It's about making sure that what we plan to use is appropriate from the start. Next is installation qualification, or IQ. Here, we confirm that the equipment has been installed correctly according to the specifications and that all components are in place. This step ensures that everything is set up properly before we start operating. Following that, we have operational qualification, or OQ. This phase tests the equipment's operational aspects. We verify that it performs consistently within specified parameters and that all functions work as intended under normal operating conditions. Finally, there's performance qualification, or PQ. This is where we validate that the equipment and systems perform effectively during actual production conditions, producing quality products that meet our standards. All of these qualification stages are part of our good manufacturing practice requirements for active pharmaceutical ingredients, or APIs. They form the foundation for a robust, compliant, and reliable manufacturing process, which is essential in delivering safe and effective medicines. This whole process ties back to our overarching goal, ensuring quality from the very beginning through proper qualification before moving into process validation. Let's take a closer look at the approaches to process validation, or PV. Essentially, PV is about gathering documented evidence that our manufacturing process, when operated within its established parameters, can reliably produce an intermediate or active pharmaceutical ingredient, API, that meets its predetermined specifications and quality attributes. Now, when we talk about the traditional approach to process validation, there are three main types we consider. First is prospective validation, which is conducted before we start the manufacturing process. It's all about planning and testing the process in advance to ensure it will perform as expected. Next is concurrent validation, where we perform validation during actual production. This allows us to monitor and confirm that the process remains within control while it's happening. Finally, there's retrospective validation, which is typically used in exceptional cases. Here, we review historical data from previous production runs to verify that the process has been consistently producing quality product over time. All of these approaches are fundamental components of GMP for active pharmaceutical ingredients, ensuring that our manufacturing processes are reliable, reproducible, and compliant with regulatory standards. This slide highlights how these strategies fit into the broader framework of quality assurance in pharmaceutical manufacturing. Let's talk about approaches to process validation, or PV, specifically retrospective validation. This method is typically used when certain conditions are met. First, we need to have clearly identified critical quality attributes and critical process parameters. These are the key factors that ensure the quality of the API. Next, it's important that appropriate in-process acceptance criteria and controls are in place. These help us monitor the process effectively during manufacturing. Additionally, retrospective validation is suitable when there haven't been significant process or product failures caused by factors other than operator errors or equipment failures that are unrelated to the equipment's suitability. In other words, if issues haven't arisen from the process itself or from unsuitable equipment, retrospective validation can be a viable approach. Another key aspect is establishing impurity profiles for the existing API. Understanding these profiles helps us ensure consistent quality across batches. When selecting batches for review, they should be representative of all batches produced during the review period, including any that failed. It's essential to choose a sufficient number of batches to demonstrate the process's consistency over time. All of these points are crucial as we aim to maintain high-quality standards for active pharmaceutical ingredients, aligning with GMP requirements. Retrospective validation, when applied correctly, can be an efficient way to confirm that our processes remain reliable and compliant. Now, let's delve into some new approaches to process validation, or PV. Traditionally, PV has been divided into 3 stages: Stage I, Stage II, and Stage III. In Stage I, we focus on process design or development, where we establish the initial parameters and understand the process thoroughly. Moving into Stage II, we move on to qualification and continuous process performance verification, ensuring that the process consistently produces quality product. Finally, in Stage III, we emphasize continued process performance verification, which is all about ongoing monitoring and improvement. What's important here is the life cycle approach that underpins these stages. Instead of thinking of validation as a one-time event, we see it as a continuous journey, an ongoing cycle of assessment and improvement. This approach aligns perfectly with good manufacturing practices for active pharmaceutical ingredients, or APIs, especially as we move into Part IV of GMP guidelines. By adopting this life cycle model, we're better equipped to maintain high standards, adapt to changes, and ensure consistent quality throughout the product's life. It's a more integrated, proactive way of managing process validation that ultimately benefits both manufacturers and patients. Let's talk about the process validation program. The number of process runs we need to perform really depends on how complex or significant the change is. For example, if we're working with complex API processes or those with longer completion times, we need to demonstrate consistency across multiple runs. This helps us ensure that the process is reliable and reproducible. Key to this is controlling and monitoring critical process parameters throughout the validation. We want to confirm that these parameters stay within predefined limits to maintain product quality. Another important aspect is verifying that the impurity profile of the API stays within the specified limits. This isn't just about meeting regulatory requirements, it's about ensuring safety and efficacy. We also compare the impurity profile to historical data or profiles established during development or for batches used in pivotal clinical and toxicological studies. This comparison helps us confirm that the process remains consistent over time and that the product quality is maintained at the desired level. All of these steps are part of our broader GMP framework for active pharmaceutical ingredients, which ensures that we consistently produce high-quality APIs that meet both regulatory standards and our internal quality benchmarks. Let's talk about the periodic review of validated systems. This involves regularly evaluating systems and processes to ensure they remain effective and compliant. The main goal here is to verify that everything is still operating in a valid manner, meaning it's still doing what it's supposed to do. Now, if there haven't been any significant changes to the system or process, and a quality review confirms that the output continues to meet its specifications, then there's usually no need to revalidate everything from scratch. This is an efficient way to maintain compliance without unnecessary repetition. Additionally, we can follow a risk-based approach in this review process. That means prioritizing resources and attention based on the potential risks associated with different systems or processes. This helps us focus on areas that could impact quality or compliance the most. Finally, this concept ties into our broader GMP principles for active pharmaceutical ingredients, especially in Part IV. Regular, targeted reviews help ensure ongoing control and assurance of quality, which is fundamental to maintaining high standards in pharmaceutical manufacturing. Let's talk about cleaning validation. The main goal here is to ensure that our cleaning procedures are effective, especially in situations or process steps where contamination or carryover could really impact the quality of the active pharmaceutical ingredient, or API. We want to focus our validation efforts on those high-risk areas to make sure we're maintaining product integrity. In early production stages, it might not always be necessary to validate cleaning processes if residues are removed later by purification steps. This can save time and resources, but only if it's justified by the process. It's also important that our cleaning validation reflects how we actually use the equipment in practice. That means considering real-world usage patterns so our validation is relevant and reliable. When selecting the material for validation, we often use representative intermediate or API samples. The selection depends on factors like solubility and how difficult it is to clean the equipment. We also need to calculate residue limits based on the API's potency, toxicity, and stability to ensure safety and quality. All of this ties into the GMP requirements for active pharmaceutical ingredients, ensuring that every step we take supports the overall quality and safety of our products. Now, let's discuss cleaning validation, which is a crucial part of ensuring product quality and patient safety. The cleaning validation protocol includes several key components. First, we need to specify the equipment involved, the procedures to be followed, and the materials used during cleaning. It's also essential to define acceptable cleaning levels, which are based on residue limits that are both practical and achievable. Monitoring parameters is another critical aspect. These are the factors we need to control and observe throughout the cleaning process to ensure consistency. Additionally, we must select appropriate analytical methods to verify cleanliness, including details like the type of samples collected, how they are collected, and properly labeled for traceability. Moving on, validated analytical methods are fundamental for accurate testing. These methods must have sufficient sensitivity to detect even tiny residues, a low detection limit, and good recovery rates to ensure the results are reliable. Residue limits should be practical, meaning they are realistic to achieve in routine operations, yet stringent enough to guarantee safety and compliance. Lastly, all of these elements come together under the GMP guidelines for active pharmaceutical ingredients, or APIs, particularly in Part IV, which emphasizes the importance of robust cleaning validation in maintaining high-quality standards across manufacturing processes. Now, let's talk about cleaning validation, which is a crucial part of ensuring product quality and safety. When we perform sampling, we can use different methods such as swabbing, rinsing, or even alternative approaches like direct extraction. The key here is that these methods need to be capable of quantitatively measuring the levels of residues that remain on equipment surfaces after cleaning. This ensures that any potential contaminants are below acceptable limits. Remember, maintaining consistent and effective cleaning processes is essential for compliance with GMP for active pharmaceutical ingredients. It's all about making sure that every piece of equipment is properly cleaned and that residues are controlled, which ultimately protects patients and supports product integrity. Let's now discuss cleaning validation, which is a crucial part of maintaining quality in pharmaceutical manufacturing. When we talk about cleaning validation, it's important to include microbiological and endotoxin testing where appropriate. This helps us ensure that no harmful residual contamination remains after cleaning. It's also essential to monitor cleaning procedures at suitable intervals after they've been validated. This ongoing monitoring confirms that the cleaning process continues to be effective during routine production. We do this through various methods, mainly analytical testing and visual examination. Visual inspection, in particular, can be very useful for detecting gross contamination, especially in small, concentrated areas. It provides a quick and effective way to verify cleanliness visually. All of these steps, microbiological testing, endotoxin detection, analytical testing, and visual inspection, are vital to ensure that our cleaning procedures are consistently effective. This ultimately helps us uphold the standards outlined in GMP for active pharmaceutical ingredients, which is fundamental to ensuring product quality and patient safety. For further support, please don't hesitate to reach out to us. We're here to help you with any questions or additional information you might need. Thank you so much for your time and attention throughout this presentation. Remember, we're committed to your success, and we're just a contact away if you need anything.